undergraduates interested in molecular and cellular genetics. And today it's my pleasure to introduce Dr. Chris Vulpe. He's our final course speaker for this class. And Dr. Chris Vulpe is coming from University of Florida, Gainesville. He is from the Center of Environmental and Human Toxicology right now, professor there. So just a short bio, his BS, well, actually in MIT, instead of BS, they say as BS. So from the legend, I was really confused. I was like, what is a BS? So he got his MD in 1986 from MIT, Cambridge. And then he pursued a PhD MD in University of California, San Francisco, and got his PhD in 1994 and MD in 1996. And after that, he started as professor in University of California, Berkeley, and he stayed there for 15 years at the Department of Nutritional Sciences and Toxicology. And in 2016, right, Dr. Vulpe moved to University of Florida. And what I want to say about Dr. Vulpe, he is a pioneer actually in the CRISPR technology, in the editing, gene editing, but specifically he is using the CRISPR for functional toxicology to identify functional target toxicity. And he worked in the wide range of organisms, started production, right, and now it's actually a mammalian. What else he told me about COVID yesterday? Yeah, during COVID, he actually used his CRISPR technology to identify novel targets specifically in response to target. Is that?
Yeah, thank you so much. Thanks a lot for inviting me, and it's been a real pleasure interacting with the students. And as I said, it's a very different kind of seminar. Usually with seminars, you're sort of forced to speak with faculty all the time with the 30 minutes, not that I don't love it, but it was really, very interesting and to, be able to talk to the students and really learn about what people are doing. So I'm going to talk to you a little bit about, maybe give you a little bit of a snapshot of some of the work that we've been doing, as mentioned, sort of applying these sort of new technologies to try to understand mechanisms of toxicity. So, I realized, coming to this department, that it's a pretty diverse background, and not as familiar with sort of the minutia of toxicology. And so I thought I'd first kind of maybe give kind of just a little bit of a global overview, just to kind of say, well, why bother with all of this gobbledygook? So one of the things that people aren't aware of is just the sheer number of chemicals that we're exposed to each day. And it's been estimated that there are about 70,000 to 100,000 chemicals that are currently being used in commerce. And that means they're being produced and used in all of the products and so forth that are around us. And that number is increasing, and it's increasing every day. It's been estimated that there are 200 to 300 chemicals, new chemical entities that are put out into the environment that we're exposed to and being manufactured. And that's every year. And this is just showing you a little estimate of how the chemical production is really outpacing even our best efforts to keep up. So we're really exposed to an enormous number of chemicals. And just for the United States, the total US chemical production and importation is 74 billion pounds. And that corresponds to, you probably heard the recent news about a single tanker blowing up and releasing some, noxious buggers out into the world. That's 10,500 miles of tankers that are produced in the United States. So the amount of exposure is significant. So, of course, being logical and reasonable people, we would not use all these chemicals and just throw them out into the environment unless they're safe. I mean, that's sort of, obvious to any, thinking person. It actually turns out, unfortunately, that in fact, that's actually not true. And most, if not almost all chemicals are put out in the environment with and expose people with essentially no information. As I was talking to the students, that's the kind of result of the sort of regulatory structure we have here in the United States. And when the first regulations came in, which was TSCA, we actually grandfathered in about 60,000 chemicals. Those are the ones we currently had. And we said, okay, well, they're being produced. They must be safe. And all those chemicals now are essentially completely unregulated. For the ones since then, there is essentially, unfortunately, still no regulation. You are really only required to do testing for pharmaceuticals, which you've probably heard of, like the FDA, and then a limited set of chemicals used in agriculture, such as pesticides. And it's been estimated that there's only even just the barest minimum of data on only 10% of chemicals, and that's any species. So the vast majority of chemicals, we just don't know squat about what they're doing and what they're going to do in the future. And what I kind of say is that we are doing the great chemical experiment of the 21st century, which is take an enormous number of chemicals, dump them on the earth, and we'll see what happens down the line. So I don't want to make people too depressed, but this is kind of showing kind of why we need approaches to understand chemicals and what their mechanisms of action are and what they're potentially doing to people. And there are ongoing efforts by the EPA, the NIEHS, and many academics to try to develop high throughput approaches, ways that we can screen these chemicals rapidly and efficiently to try to understand their mechanisms of action. Now, my work that we do is a very, very teeny little piece of that, which is kind of a specialized area of trying to understand what is the mechanism of an action of a chemical. How is a chemical actually causing the adverse effects that you see in people or in any species? So what is the approach that we use? So I'm going to kind of say the same thing multiple times because sometimes this can be a little bit challenging to understand, especially for people that aren't familiar with it. But hopefully by the time I've said it, 4 times, hopefully it will make a little bit more sense. But essentially, the approach that we're using, as I said, is to try to understand what are the molecular mechanisms of toxicity. So how do we do this? Well, we take advantage of CRISPR technology to try to target individual genes, but we do this in a sort of massively parallel way where you can actually screen multiple mutations across and simultaneously assess the effect of multiple mutations at the same time on their response to a toxicant. And then you can look through those mutants to try to identify the ones that are important. So essentially, we're using kind of a screening approach. We create a whole bunch of mutants, and then we screen through the mutants to try to identify the mutations that are important. Now, this approach is not unique to toxicology. This approach can be used for any question that you would like to answer in terms of a particular phenotype. And it's been applied to bacteria, to mammalian systems, to plants, even to yeast. So, I mean, you can use it for anything. And so, although what we're doing specifically may have no particular relevance to your own work, I think it does, the sort of general approach can be potentially applicable. So what we're doing, and we kind of grandiose, I don't know how to, with bigly, is that a good word? We've decided, we call it functional toxicology. And so this isn't really a new idea. And as I was telling the graduate students, this is basically genetics 101. It's what we've been doing forever, right? We create mutations, right? We screen through those mutations to find a phenotype that we're interested in. And the only thing that's different now is that, we can now do this in a much more organized and less random fashion by using CRISPR. And the idea is not, it's pretty obvious, which is if you mess up a gene, and if you muck it up, and that affects the role or how it responds to toxicity, that gene is probably important because you mucked it up and now suddenly you've changed how the organism or cell has responded. It's either more toxic or it's less toxic. And, sort of the corollary to that is if you mess up a gene and it doesn't do anything, at least as related to toxicity, then it's probably not that important in the organismal response. It's not saying it's not important, it's just saying it's not important for toxicity. And so with functional profiling, what we can do is we can actually now go through these and systematically test multiple genes. Whereas previously we kind of relied on random chance, and it was really pretty laborious to go through and identify individual mutants and test them. It's the basis of most of our understanding of biology, and CRISPR has just made this a lot easier. And so, what we can do is you can target an individual gene, disrupt its function, and then ask about its toxicity. And then we can now identify, by doing this systematically for a whole bunch of different genes, what are the functionally relevant processes and pathways involved in this process. So I want to sort of give you an overview for people who aren't used to thinking about toxicology, about like what is it that we're looking for. Well, when you think about a toxicant, it's essentially, and this is on a cellular level, and you're thinking about the mechanism of toxicity, you're thinking about things that are pretty obvious. Well, how is a toxicant getting into the cell? So most toxicants have, if they don't just diffuse across the membrane, they have to get in. It turns out many toxicants actually hijack our nutrient transport systems to get in. And so we've evolved to use them, and the toxicants come along and say, "Ooh, that's a good system to use," and we'll go in that way. But we don't know what those are. So there's an import step. Once it gets into the cell, it also has to transport around and move around. And again, it's usually going to hijack something that already exists there. And as you're probably aware, many toxicants are metabolized, and they can either be metabolized to be made less active or more active. And actually, when you think about it, you can often, an individual toxicant will sometimes be metabolized kind of like what I would call a shotgun spray of metabolites. They make a whole bunch of different metabolites, and unfortunately, all those metabolites potentially have different effects. So it can be kind of challenging. And, sort of simplistically, you can sort of think of these toxicants. They can either be direct acting, which means they're not metabolized. They interact with some target, and that leads ultimately to the bad things, or what we call adverse outcomes in the toxicology field. Now, it also could be the metabolite that's involved, and then it, again, has another target. And, the cell is responding to this, for instance, turning on cellular response pathways, damage repair pathways. And by looking at some of these things, you can actually get clues as to what the toxicant is doing, right? Because by understanding this, you can get a sense of that. And then ultimately, often many cells and organisms try to get the thing the hell out of there, and so they have export mechanisms. So when you think about a toxicant, these are the kind of questions you're trying to answer. And unfortunately, for the vast majority of toxicants, we don't know any of this. And if we know anything, we may know metabolism, because that tends to be something that's most studied, but we only often know it on a very rudimentary scale. So this is just an example of how CRISPR are knocking out things. So as you can imagine, if you knock out the gene involved in import, simplistically now the thing doesn't get in, so your toxicity goes down. Conversely, if you now knock out something involved in response to a toxicant, like a cellular stress response, okay, now you don't have this cellular stress response, so suddenly now you've increased your outcome. So this is the kind of sort of rationale that we have for looking at these and doing these studies and giving you a kind of a background. So how do we do it? Well, targeted CRISPR is probably something that most now graduate students and undergraduates are familiar with. And essentially with CRISPR, and most of the things that people are aware of, are what I would call individual CRISPR. And so, what's amazingly cool about this system is that essentially you can, if you have a target sgRNA that's complementary to the DNA that you're trying to target, you can essentially target any DNA in any organism if you can do it, if you have the sequence. And then it will go into the cell, and this Cas9 is relentless, and it will just keep cutting the DNA until the natural DNA repair mechanisms of the cell or organism mess up. They make a mistake, Cas9 no longer recognizes it, and you get a mutation. Importantly, and that's I'll say it, it's kind of a random mutation, and it's not, when you say Cas9 is precise, it's precise in where it targets, but it's not precise in the mutation that's put there. It's a array of mutations, and it's DNA site, it's that specific to each chromosome, each allele. So if you did this in corn, you're gonna have 6 or however many alleles you would have. But it's very highly efficient, so it will go and often target all the alleles. So, so that's kind of how you do it. And so you can do this and for individual knockouts, and that's fine. But to do all of this for the about 18,000 genes in a human is obviously pretty tedious and would take a long time because you generate 18,000 individual clones. So how do we do this for screening? Well, essentially in screening, we're doing exactly the same thing, but what we do is we do it in such a way in a cell culture, an organism, where we introduce into a cell just a single sgRNA so that it targets just one gene in a cell. But we do it in parallel such that this cell we target this gene, this cell we target this gene, this cell we target this gene, and so on, all the way out to the 18,000 genes. And what you end up with is a pool of mutants. So this is the pool of mutants like we would have made before just by using, radiation or whatever. But now these are unique targeted mutations going after each individual gene. And furthermore, as I'll explain to you later, they've actually been flagged. So we can follow these mutations over time and identify which ones are important. So how do we do that? And we take advantage of a system where it's an integrating system. And this is a vector system that expresses both the sgRNA and a Cas9 and a selectable marker. It integrates into the genome and it targets the gene corresponding to the sgRNA. And so this tells you that this cell it's knocked out, and you can identify that knockout by the sgRNA that's tagged. And how do you figure out then the genes that are interested? Well, what you do is you just PCR up the sgRNAs and you count them. So I'm going to now just to give you an explanation of how you make these libraries. So generally, you can go on any gene set that you're interested in. This could be a whole genome, and you can do this for anything. We've made like bunches of these libraries. It's actually very easy and very straightforward. It's just cloning, and you can clone anything you want. And you can go after a whole genome or a targeted subset. You pool them as an oligonucleotide, and you make this pooled plasmid library. And then you package it into a specific virus, can be almost anything, but the one that's most commonly used is a lentivirus. And every sgRNA is targeting a different gene. You usually have multiple sgRNAs for each gene so that you have a certain amount of redundancy. And in the all-in-one systems, again, you've got a one vector system that integrates. So what do you do now in these screens? Well, you may take your library, you introduce it into the lentivirus, and then you do, this is the real trick, which is essentially you do a low multiplicity of infection. And so what you're doing is you're introducing less than one virus per cell. So the most that any one cell can get is one virus. And by doing that, you now have individual clones which have a knockout of a single gene. You get rid of all of the ones that don't have it by doing the selection. And now again, you have your library of mutants. And generally, we like to have multiple copies of each knockout to get what we call coverage, just to be able to ensure that we can see differences in a statistically robust way. So then what do? Well, essentially now you've got your library and now you just abuse it. So you take your library, and here just showing you an example, and you don't do anything to it. And so this is the number of each of the cells of the mutants, and you just grow them, and in an ideal situation, they would all stay the same. But in the case of a toxicant, you can see here, this guy, after you beat it with a toxicant, it's that mutant goes away, so there's less of it. This guy says, hooray, toxicant, I'm happy, I'm gonna grow like crazy. This one says, toxicant, I don't care, right? So now you have essentially, you've got a readout here, which is telling you whether it's sensitive or resistant to the toxicant. So how do you read that? Well, you read it just by simply making genomic DNA from these preps. You amplify up the sgRNAs using these PCRs, and you just count them. You just literally count the sgRNAs, and as you'll see you get no change. There's the same number of sgRNA barcodes. You'll see resistant ones where there's now more of that barcode, and then sensitive ones where there's less of that barcode. And then you can use computational analysis just like you do for RNA-seq to pull out the genes. But what you're measuring here is not RNA, you're measuring the number and the quantity. So I'm going to give you, whoops, so it looks like I messed, one of them is gone. But I was going to give you two examples of genome-wide screening and one targeted. Now the two examples are arsenic trioxide and dieldrin, and the one targeted one will be a surprise because it looks like it did not get into my final thing, but I'll give you a clue. It was with a collaboration with faculty member here at the University of Kentucky. So I'm going to talk briefly about these. So we've now done this on a wide variety of different toxicants, and in the class that Olga mentioned, they actually went through two papers from our lab. And when I asked the class, I asked them, well, what would you prefer? Would you prefer I talk about something you've already read, or would you like me to talk about something I haven't read? Well, you haven't read. I'm not sure. I think I've read my papers, but I'm not entirely sure. But anyway, so I asked them, and they, of course, I had done my talk, and of course, I thought, oh, well, that would be a really great idea. I'll focus on what they've done, and that would be something. And I was thinking about it, but that's goddamn boring because they just already read about this. Why do they want to see anything? So anyway, let's just say I redid my talk to focus on things that I hadn't been. So this is hopefully new for the people in the class. Okay, so I'm going to talk specifically about arsenic and dieldrin and the work that we've done on those. So arsenic was actually one of our very first studies. Now we did this on arsenic trioxide. So this was kind of a dual study, and the arsenic trioxide is also used as a drug for the treatment of some types of cancer, and it's also a very well-known toxicant and affects literally millions and millions of people. Both here in the U.S. and in Bangladesh and India and other countries, because arsenic is present in the groundwater from these places, also in Chile. So it's a very important toxicant. Unfortunately, it's still the mechanism of action of it is still very uncertain, and as I often say, it really depends on who you ask. I'm not sure that we added anything to that, but we'll go through what we identified. So we did a sort of standard now study, and one of the things I'll say is that we use sort of an intermediate dose of arsenic, which is around the IC30. And the reason why we do this is essentially what we're trying to do is we're trying to find a concentration that doesn't, the sort of intermediate between, so that we can identify both things that are sensitive and are resistant. So if we were just interested in resistant ones, we could use really high concentrations. You kill everything that's sensitive, you kill everything, right? But it's very difficult to identify anything that's more sensitive because you've killed pretty much everything. And the same thing is if you use a really low concentration, you'll really identify the sensitive ones, but it's really hard to identify resistant ones because everything is resistant compared to that low dose. So we kind of use this intermediary dose as a way to hopefully get sort of both ends of the spectrum in terms of sensitivity and resistance. So what does the data look like? Well, the data looks kind of like what you're used to with RNA-seq. Anyone who's done an RNA-seq experiment, you'll say, well, this looks very similar. And it's essentially you're again comparing the control versus the arsenic exposed. But instead of each of these dots representing an individual gene or transcript, they represent each individual sgRNA. And above the line here, this is a fold change analysis, and then here are the counts. And again, things that are resistant now are going to be above the sort of log, the zero line, which essentially means there's no difference between arsenic and control. So resistant ones are up here, sensitive ones are down here. The only difference is that there are multiple sgRNAs for each. So that adds a certain amount of complexity in your analysis. So we did this and we identified an initial set of mutants, and we decided to go ahead and make a custom validation library. So we actually we were doing several screens at the same time, and we combined the results from all of the screens and actually made now a custom library where we just made a targeted library focusing on those genes plus a bunch of unrelated genes, and just re-screened it to identify again. And then so we essentially did a confirmatory analysis and identified now a subset of some of the genes that we identified initially that were consistently resistant and sensitive. And this is the demonstration of the results, and I'll show you kind of how it looks. So you have the gene name here. So the fold change is essentially, as you could imagine, this is the comparison of how prevalent this mutation is in the treated versus the control, right? So there's 5.294 times more of this mutant in the treated as in the control. So this is a resistant one. And this then shows you the number of sgRNAs that we tested that showed that effect. So all the sgRNAs that we used to target KEAP1 show the same effect. And then this shows you a Q value. And as you can see, the Q values are kind of insane, which essentially just means that these are really statistically significant. And so you now identify these individual genes, but you have the same sort of issue that you have then when you're dealing with RNA-seq. How do you make sense of this information, and what does it mean? And we use the same sorts of tools that are used for RNA-seq, which is essentially kind of pathway analysis, enrichment, gene ontology, enrichment analysis, other sorts of tools, computational tools, which basically try to help us understand what are the biologic processes and functions that are enriched or overrepresented in our set of genes that we've identified. And so you can sort of see here that kind of showing you kind of some of the things that you identified. And so we identified, selenocysteine, oxidative stress, cellular damage. So some of these things we were expecting. So oxidative stress, that seemed like something that made sense. Cellular damage response, that makes sense. So what the hell is selenocysteine? We were just like, okay, we had no idea. Because selenocysteine, for people I'll talk about it later, but it's usually involved in actually stress response, but these were guys that you were actually resistant. So this was contrary to what we expected. We expected if you knock out selenocysteine metabolism, you should make things less because it's involved in oxidative stress response. And it just said oxidative stress response. But so anyway, it was completely confusing. So we were like, okay. So, and then we identified also in the, some things too, transporters and a bunch of other things. So it's still a challenge to be able to make sense of it. But I'm going to pull out a few little tidbits from it. So KEAP1, which was the most resistant, kind of makes sense because KEAP1 is actually a repressor of one of the most common, whoops, I have a misprint here, that should be NRF2 there. So it inhibits NRF2, which is an antioxidant, which is the primary transcription factor involved in turning on the antioxidant response pathway in mammalian system. And homologs are common across multiple different species. So this turns on a whole array of things involved in response. And that's normally regulated by KEAP1, which keeps it in the cytoplasm. And when it binds an electrophilic or oxidative stress, it releases NRF2, it goes into the nucleus and turns on a whole bunch of things. Now, it's not, not surprising because we kind of had a sense that arsenic causes oxidative stress, although there's a lot of controversy about what its relevant importance is. This suggests, at least in our system, that that it is particularly important. So that made sense. And then we identified some transport processes, and that wasn't as clear. But when we started looking in the literature, we identified this aquaporin 3 as being resistant, and we were like, well, that doesn't make a whole lot of sense. But when we looked in the literature, it turns out other people had actually found that aquaporin 3 was involved in arsenic transport. So that was cool. And then again, we identified a gene called ADCC1 or MRP1, and again, that was already known. So again, within the caveat that this isn't this one cell line, it was consistent. We also identified a zinc transporter, and to be honest, we have absolutely no idea what the hell the zinc transporter does or why it has anything to do with arsenic. And if anybody has any suggestions, be interested to know, but it's a pretty strong candidate. So what about this selenocysteine stuff? So what was, as I said, it's the 21st amino acid, and it's been previously known that arsenic binds selenium. And what was really confusing is that we found that essentially any gene in this selenocysteine biosynthetic pathway made the cell, resistant. So it wasn't like something we could say, oh, well, it's just, I don't know, one of these genes, and maybe it's some sort of weird, mutant that we identified. It's like hitting us over the head. It's like, no, it's not just this gene, it's any gene in this entire pathway. If you knock it out, makes the cell. And so we're like, okay, this probably is real. So now we have to understand what the heck it means. And as I said, it's very confusing because all these selenocysteine proteins are involved in response to oxidative stress. So you get rid of them and now you're making them resistant. So yeah, this was a challenge. And so we've come up with two hypotheses to explain it. Honestly, we don't know which is the right hypotheses, but I'll just go through them. So it actually turns out that it's been known for a long time that selenium and arsenic actually can form a physical complex. And it actually comes from some initial studies in ruminant animals who are grazing on selenium-rich soil versus arsenic-rich soil. And it was found that if you gave selenium to animals who are on arsenic-rich soil, you could protect them from arsenic poisoning, and vice versa. Although I would not recommend this for you to try at home. If you if you have selenium toxicity, if you take arsenic, you actually protect against selenium poisoning. And it's because it actually forms an insoluble complex in the gut of these animals. And so we thought, well, could this be happening in an intracellular level? Could this be happening inside the cell? And so we gave that a try. We actually added arsenic, and it does seem to, it's kind of modest, but it does seem that if you add, selenium, you can actually sort of mitigate the toxicity. So the idea here is essentially that by mucking up selenobiosynthetic processes, we're increasing the available pool of selenium in the cell, which can then now bind to arsenic and make it unbioavailable. So that was our first hypothesis. I'm not entirely sure whether, this is clear. The second hypothesis was actually when I was calling around people who worked on selenocysteine proteins saying, hell, I don't know what the heck this means. What, do you have any ideas? And so I talked to this guy in Vermont, Robert J. Hyundai, and he told me that in fact, it turns out that one of the proteins that's involved is thioredoxin reductase, which has a selenocysteine actually is kind of has a double-edged sword in that it turns out that arsenic trioxide has been previously shown to bind to this and to inactivate the reductive activity. Okay, so that's fine. But what happens is when you inactivate the reductase activity, it actually activates another domain, an N-terminal NADPH oxidase activity. And so it's this strange thing that when you get rid of one, you turn on the other. And so what could be happening, we have no direct evidence, is that by preventing the production and the putting of the selenocysteine here, that we're actually now activating this ROS generator here in thioredoxin reductase. And so this is kind of our other hypothesis to try to explain that paradoxically, we are creating a pro-oxidant. So that's kind of where we are on this. And we've identified now these processes that are involved. And so, what was our conclusion from this? Well, it was oxidative stress is a major player, disrupting selenocysteine. The mechanism is really not clear. There was arsenic transport, and there's something going on with zinc. And so it was, it was very interesting to us, and then we identified now a series of different genes and pathways that were involved. Okay, so sort of one example, one of our first examples, and I'll kind of go on to tell a little bit now about a different example, which we then tried to apply this to a more, initially we done on a K562 cell, so we thought we would now see what could this work in any sort of other cell systems. We worked on a neuronal system, and we focused on a compound called dieldrin. So dieldrin is a organochlorine pesticide, which luckily is now banned, but unfortunately, it will be here until the sun goes supernova because it is essentially does not degrade, and it's still highly contaminated in many agricultural sites across the U.S. And in Florida. And there's a particular place you've probably heard of, like Apopka, where there's actually a ton of this compound. Now, it's been epidemiologically linked to an increased risk of developing Parkinson's disease, primarily in agricultural workers and people who use who were pesticide appliers. So, and it turns out that it actually accumulates in the substantia nigra, which is the part of the brain that's affected by that, but its mechanism of toxicity is not known. And so, in a collaboration with Bin Liu at UF, and a graduate student in his lab in Max Russo, we started a project to try to see whether could we use a genome-wide CRISPR screen to try to answer that. So we did a screen here now in a dopaminergic neuronal cell line, which is probably one that some people may be familiar with, which is an SHSY5Y. Now it's a tumor cell line with all of those caveats. And so we did the kind of approach very similar to what you saw there and identified a series of genes that were involved in response to dieldrin, both sensitive genes and resistant genes. And when you looked at those sets of genes, we identified then specific pathways that seemed to be enriched, that were identified from this analysis using different types of pools. And what was particularly interesting to us was that we were identified genes involved in these pathways, the ubiquitin and autophagy and mitophagy especially, that were involved, that seemed to be involved in response to this dieldrin. But what was interesting was that in fact, these are the genes that have been implicated previously in genetic forms of Parkinson's disease. And this was the first indication that I know of sort of, because there's like what I should have said is that there is a significant proportion of Parkinson's disease, which has a genetic component. But the vast majority of Parkinson's disease is this sort of unknown etiology. We don't know what causes it. Many of the forms of Parkinson's disease, probably ones you're familiar with, are earlier onset than the late onset. And so what was cool for us is that we suddenly, going into this very quietly, my battery died, going into it very quietly, we were surprised that we actually now saw these genes that were involved in,
They won't be able to hear you on Zoom.
oh, okay.
You can turn my microphone. Is that one working?
This, you want me to carry this one around?
No.
Okay, now I will be a very formal person.
Is it
Yeah.
not?
you might-
Yeah, so anyway, so that was what was cool. So we were like, wow, that's kind of interesting. And in fact, one of the major sort of, I guess what you would call it, kind of a hub, was centered on PARK2. So PARK2 is called Parkin, and this is actually one of the genes very well known as being an important player in the development of Parkinson's disease, and appears to be involved in the regulation of mitophagy, which is a breakdown of mitochondria. And similarly, we also identified a whole series of genes involved in the ubiquitin pathway. And again, these are ones that have been previously identified. And I just will kind of show the work identifying both autophagy and mitophagy, and sort of confirmatory analysis to demonstrate that in fact we were seeing differential effects with it. And then again here showing the sorts of pathways that were involved, which seem to center around mTOR signaling and the PARK2, one being involved in resistance and the other being involved in sensitivity. And then again with the autophagy pathways sort of downstream in a way to both of them. So based on that, we came up with sort of a model of, suggesting that dieldrin potentially is sort of interacting in modulating, something involved with mTOR, which potentially then is going downstream to both the autophagy and mitophagy pathways and the ubiquitin protease system. And, the question, of course, is, how does that ultimately result in Parkinson's disease? Well, that's a question for later. But I think it did suggest that there is a potential similarity between the genetic forms of the disease and the environmental forms of the disease, at least in the biologic pathways that are affected. And so that was kind of interesting to us. So the last thing I wanted to talk about was a collaboration here at the University of Kentucky. And the idea here was focused on, we decided to use a subset library. And this was a targeted library where we pulled out genes that were specifically related to toxicology. There are about 3,675. I won't go into the details of it, but these were a set that were identified not by us, but by many other experts in the field, and thought that this would be a useful tool and enable more rapid screening. So one of the things I didn't mention in this thing is it requires large numbers of cells to do these screens. And when you're doing it on a cell that grows in suspension, that's not so bad. You just grow more cells. When you're growing them like the SHSY5Y, and you have to grow 30 million cells, and you have to grow, 3 to 5 replicates of 30 million cells, we're talking, you're talking about the number of incubators that you use, not the number of flasks. So, poor Max was growing, just enough to feed a small army. And the amount of fetal calf serum that we use was somewhat large. So we thought, well, we need sort of more effective ways to do this. And so we working on this, and then I was contacted by Dr. Awa. I always mess up your name.
Awa, yeah.
Awa. Yeah, so this is not him particular. I have, I don't know what to call it. It's like this disorder and inability to pronounce things. I inherited it from my mother, so it's probably mitochondrial. But for the whole time that I was in the Bay Area, I could never, I was always like, okay, is it Marin or Marin? Marin. I still don't know, but hopefully someday I will. But anyway, Dr. Oh crap, you just told me and I can't even remember. He contacted me and, and this was actually, he read some of my yeast papers. First of all, he actually read one of my papers. So I was like, yeah, that was great. And then, I said, well, because we had done something similar in yeast before, kind of the same idea, kind of using yeast. And I said, well, we're doing this new fancy CRISPR, gobbledygook. Would you be interested in trying that? And, and maybe to his chagrin, he said yes. And so we started a collaboration, and he had his lab focuses on developing novel chemotherapeutics. He had identified multiple different gold-based compounds. Here's one here called JHK21, which I now know stands for John Jim, which I didn't know at the time. I was always like, what the heck is this? But anyway, so and this was just an example of one of the compounds that he's created. And they seem to be potentially effective. But again, it was the same question. What is it doing? How is it doing it? And so we then applied this CRISPR screen using what you see now as sort of the approach that's typical. We said, well, let's just screen and see what we identify. And so we did that, and we started going through the genes here. And I don't know if anybody, looks at genes. We started looking at these, and we were like, huh, that's interesting. The first one we identified is ATP synthesis. So this is making, this is the ATP synthesis, the mitochondrial ATP synthesis. Okay, well, that's interesting. Superoxide dismutase. Some people may not know that that's primarily mitochondrial. ATP, ABC, well, that doesn't count. Cellular, whoa, that's involved in electron transport. Oh, mitochondria. So we were like, whoa, that it was, again, one of these cases where the data just hits you over the head and says, hey, you probably should think about the mitochondria. Not. And so that's kind of what we did. And we also identified a series of resistant ones, which were not as interesting, but there's actually probably a lot of information that we can pull out of it. So a lot of work here, all of the work I'm going to talk about comes from his laboratory after this. We were just the CRISPR people. We handed off the results to him, and they've done an incredible job in following up on all of this. And essentially, as I said, this is showing that a whole bunch of hits seem to be involved in the mitochondria. They did some initial work when you do individual knockouts to confirm it. They did this transporter here, as well as this resistant gene. And, people like these because it's like, we did this fancy screen, is it really true? And so you like to confirm. And as you saw previously, initially, we would do a whole secondary screen and so forth. I think it's gotten to the point now where people are more accepting of it. So anyway, that's kind of what we, what they did. They confirmed it, and in fact showed, like in the ABC knockout, here, this is involved in export. They actually saw that the cellular levels of this drug increased, again, showing its importance. So what's cool then is that after they identified the mitochondria, then they said, well, it looks like it's affecting the mitochondria. Can we do further work to study that? And so then first they looked at mutants, and they measured using this fancy thing that some people may be aware of, which is called the seahorse, which is essentially a way of measuring mitochondrial function in a live cell. And they measured the oxygen consumption and the extracellular acidification rate. And they did both first the ratio of mitochondrial to glycolysis. Here's the control. And I think you can see here for several of the knockouts that there's an effect on that. And then they did a mitochondrial stress test, and that further, again, showing that in the presence of the drug, you really mess up respiration in the mitochondria. So direct evidence that it's actually affecting the mitochondria. So this was really exciting evidence. And then they did a further study to try to understand what it's doing. And they used a dye that detects changes in mitochondrial membrane potential. It's called JC1. And it's kind of cool because it's the ratio of green to red, and it depends on the mitochondrial membrane potential. And again, showed, you can appreciate it to a certain extent here that there's more of the green. But when you do it now in a sensitive strain, you can really now appreciate how much more sensitive it is and its ability. So this is kind of an example, I think a good example of how you can go from these CRISPR screens to now going down to a real molecular mechanism of understanding what a drug or a chemical is doing. And I was very happy that I was able to collaborate with Dr. Law on this study. And so I think it really helped us identify genes involved in mitochondrial function, show that it seems to be involved in membrane potential, and it's a novel mechanism of action for chemotherapeutics, and that there's considerable translation potential. And Dr. Loya has, similar drugs that are being, developed with potentially similar mechanisms of action, which actually could be useful in the treatment of different forms of cancer. So to finish, I kind of just want to give you kind of an overview of what we think you can learn from CRISPR and from functional profiling. And so I think one of the most useful is that it helps you identify the causal genes. So I'm making a jab at what I call correlative omics. So this is a lot of omics technologies such as gene expression, proteomics, even metabolomics. You get a lot of changes, but it's really hard to know cause and effect. And I think one of the cool things about this approach is essentially it's really causal. You knocked out the dang gene, that gene is gone, and you're seeing an effect. Therefore, that gene is important. With other approaches, you got all these things up and down, and you don't know. It's really hard to say that it's actually right. So I would, say that's involved. I think the other thing is that it gives you information, not just on whether it's important or not, but it actually gives you relative information. It ranks the genes, which is kind of cool. It says this is really, really important. You knock this out and you muck with it, and you really make yourself sensitive. And that helps you kind of rank things, because when you do them one by one, okay, and this has actually turned out to be true in many mouse systems, you'll see changes. You'll see changes. You knock out a gene, oh yeah, that's sensitive to Tylenol, right? You knock out another gene, oh, they're sensitive to Tylenol. You knock out another gene, oh, and you're just like, suddenly you're like, well, every damn thing I knock out messes with thing, and you're like, well, which one's the real one? And you don't know. And I think that's what's cool about this is it ranks it for you. It tells you, hey, this one's really important, and you should pay attention to that. So anyway, I think that's cool. And then finally, and then my ultimate goal is that I think the more we do this, we're going to start to converge on a series of sort of common response pathways that are going to help us understand how cells respond to toxins. We know a lot about that already, stress response, but the more we do this, I think the more we realize, well, we don't know a lot, because as you saw, there's a lot of genes that we're like, what the hell does this gene have to do with anything? And we start seeing the same things over and over again, which leads us to start questioning, well, what are these genes? What are they doing? And how do they actually be involved in stress response? That's my hope, is that over time, we're going to start to make an integrated picture. So there's a lot of caveats though with this, and I want to make it clear that there's some serious issues with this approach. So first of all, we're talking about a single cell. We're talking about a mucked up cell, which is a cancer cell in many cases. And you have all that kind of caveats. Most of these cells don't have active metabolism. So you haven't taken into account the metabolism. The obvious thing, we're doing this in a cell. That doesn't say anything about an organism. It only tells you about that cell. So there's a lot of problems with that. It's a limited snapshot at a particular dose in time. Is it relevant to low dose? I don't know. Is it relevant over time? I don't know. So, I mean, there's a lot of problems with this, but it does give a window. And it doesn't incorporate toxicokinetics. So toxicokinetics is how the thing gets in there, how it gets out. It doesn't give you any information on that. So there's a lot of things you're not getting. And I think that it's important to, that just because it's important for a cell doesn't mean it's the most important for an organism. So you really have to confirm this with in vivo functional studies, which is one of the things I've been talking with Olga about. Could you use potential other model systems to actually now test these things? You identify it in a cell, now you need to test it in a whole organism. It's a pain in the ass still to do it in a mouse, but maybe you could use cell ligands or some other easier system, or not easier. No, I don't know. Okay, I'm gonna say faster, better, a better organism for your study. Okay, so those are some of the caveats. Where are we going from here? Well, I won't go into all of these things, but I'll just give you a quick thing about how we're hoping to use this now to understand human genetic variation. And this is what we call our tox-bear CRISPR. And this is a targeted functional approach to try to understand the role of genetic variation in response to toxicity in people. We all vary in our response to a toxicant. Some people, for every chemical, there is an S-shaped curve, or however you want to put it, a distribution of response. And unfortunately, we don't understand that currently. We don't know why some people develop disease in response to toxicant and other people do. And the approach that a lot of people have used in the past is to do what's called genome-wide association studies, where you try to look between an association between variants in all the human genes and the development of the disease. But that's a very challenging approach because, one, you have to have a large population of people, and you have to have exposed people, and you have to have people who are exposed to only one chemical, right? But we're not. As you saw, we're being bathed in a stew of chemicals, right? So we're exposed to a whole bunch of different things. And so it's really challenging to sort this out. And so what we just thought would be a useful idea, and luckily the NIH agreed with us, was let's focus on the set of genes which are commonly mutant in people. So it turns out there's about 1,500 genes in people where there are loss of function mutations in the gene are common. And we define common arbitrarily as a mean allele frequency, or basically greater than 0.1% across all populations. That may not sound pretty rare, but if you deal with millions of people, it's a lot of people. So what we're doing essentially is we're saying, okay, these are the things that are important or could be important, or at least no, okay, they're not saying anything about important, sorry. These are the things that are common. So now the question is, okay, they're common. So now if they're actually out there in the population, what do they do to toxicity? Do they do anything? Right, or something. And so what we're doing now is we're screening these guys using the same approach to try to identify the genes that are involved in toxicity response. Because then that will tell us, okay, of the genes that are mutant in the human population, which are the ones that we should actually be looking at, and looking at whether people have that variation or not, and we should be concerned about it. At least that's the idea. I don't know if it's going to work, but that's kind of where we are. So finally, I just want to thank the enormous number of people who did all this work, certainly not me, the graduate student, Amin, who started down this path. Manny is a long-term scientist in my lab. Max worked in Vin's lab. Alex is a biostatistician. Dr. Awa. So yeah, we worked here. These these are collaborators on a project I didn't talk about. Keith was involved in the Toxcaster. These are biostatisticians that I worked with at Berkeley. And Vulpe is my longtime collaborator. Young Zhou is a graduate student who came to my lab to do the CRISPR. Martin Smith is the Superfund director at Berkeley who started me down this path. Alex is the long-suffering biostatistician. And with that, I'd like to take any questions, and I apologize. I realize I've gone 5 minutes over time, but thank you for your attention and happy to take questions.
Question?
Thanks a lot for that. That was super interesting and I have like a ton of questions, but
Okay.
I'll just offer a comment and a question. So
Okay.
for the zinc thing with arsenic, there's quite a bit of literature on arsenite finding the zinc finger proteins.
Oh, that's interesting.
And then,
Oh, oh, I see what you're saying. So
Yeah.
it's possible that by increasing the intracellular zinc concentration, that you could shift the equilibrium with those zinc proteins so that the arsenic doesn't bind. And so when you're in a situation with excess zinc, yeah, okay, I see. So if you're in
Yeah.
a zinc deprivation state, then the arsenic could come in and displace the zinc fingers. That's a really cool
yeah, there's
idea.
a bunch of papers on
Wow.
that.
I have no idea how to test that, but that's a cool idea.
And then for the selenocysteine one, the thing that came to mind and kind of raised a question for me about pleiotropic effects of these knockouts.
Yeah.
So for both arsenic and selenium, the detoxification involves methylation.
Mm-hmm.
And I wonder if you're reducing selenocysteine production, it may be down regulating the methylation. I don't know if it's the same genes involved in methylation of those two. But
I honestly don't know the answer to that question. I'll certainly look into it. But that's, yeah, we didn't consider that. And yeah, I don't know. I mean, that's kind of in some ways what we hope is that we put this stuff out there and then some people who are smarter than us will actually figure out what the heck is going on. But yeah, that sounds like the hypothesis that we could test. So we could maybe look into that and see. Yeah, if you push the literature to me, I'd be really interested in that.
sure. Yeah, I mean, I guess just in general with these things, even with like regular mutants, you often wonder, are you really just affecting that gene?
Oh, yeah, for sure. I mean, yeah, there are all the downstream, effects, and it's hard to know, yeah, what is actually going on. The only thing I would say that would maybe argue against that is that, well, okay, I'll look at the data again. I don't recall seeing things involved in methylation in our set. So normally, like if it's a downstream thing, then we will often identify those downstream players as well somewhere in our data. But often, sometimes you don't, there's so much data that sometimes if you're not looking for it, you may not see it. I don't recall it coming up as like an enriched process or anything like that. But, that doesn't necessarily mean anything.
Chris, do you use the libraries when you do those mutations?
Uh-huh.
Are those partial knockouts or complete knockouts?
Okay, well, so as I mentioned at the beginning, so CRISPR is despite it's being called a precise, is actually very imprecise. And so when you make those mutant pools, so as I said, we like to have a pool of 500 cells per sgRNA. And then we have multiple sgRNAs for each gene. So when every cell of those 500 of each sgRNA is going to have a different mutation in the cell, in each allele for that gene. They are designed, the sgRNA, and I think I was talking to students about, are computationally identified and are predicted to lead to disruption of function, but it's a random process. So I would say that it's really, it's more equivalent to kind of you're really getting it's not like an RNAi where you get a steady decrease across all levels. This is actually, in some percentage of your cells, the mutation is going to do nothing. And in other cells, it's going to have, you're gonna knock it out completely. And in others, you maybe are mitigating it. So you're getting kind of a spectrum of effect. And the hope is that by sampling enough of them, that you can see an effect. So it is an imprecise process.
You mentioned in conversation if you sequence them, so you can identify exactly the mutation that was.
So you can do that. In practice, people don't do that because, you know, obviously you've got 18,000 genes. It's a hell of a lot, you know.
No, just the candidate, like.
Yeah, you could, but usually what you do is you use these screening processes and then you do, you're particularly interested in do a confirmatory analysis where I think some of those things you may saw, you validate how much the knockdown, how much knockout you get. And usually what you'll do is you'll screen through multiple different clones to identify one that leads to a complete knockout. Although what's kind of interesting too with this is that it also gives you the ability sometimes to identify even genes that are essential. Because essentially, if you knock it out and it actually kills the cell, it will be gone from the population. But it'll be gone from the control population and the treated population. And then now if these hypomorphs are even now more sensitive, you can potentially pull them out of the analysis as well. So that's kind of that's a sort of convoluted way of saying that we can take advantage of the fact that it's an imprecise mechanism, an imprecise process. But to answer your question, it is a random assortment of things that are supposed to be targeting and mutating that gene. Now there is an effort, as I mentioned, to try to come up with an annotated, collated set of sgRNA which are known to be functional, at least for like mammalian systems. When you do this in another species that maybe hasn't been as much work on, again, it's always a computational effort. One of the approaches, though, that people have used is to do what's called domain targeting. And the idea here is to have your sgRNAs go after conserved functional domains in the protein, so that if you do target that region, you have an increased likelihood that it leads to a functional defect. Now, none of these use that approach, but as I mentioned, when we did the SARS work, we created a new library and we actually made a domain targeted one. We're actually now, for this ToxBear process, are in the process of trying to functionally validate that every sgRNA does what it says it does, because we're going to be using this limited set of 1,500 genes. We're going to have a reduced number of sgRNAs, and we want to really be assured that those sgRNAs are not going to be targeted as we think we are. And so we're in the process now of trying to test that. It's actually a pretty difficult problem because you're trying to identify single mutant genes in all of the non-mutant genes. So let's just say it's NovaSeq, times 6 to actually get the number of sequence. I don't even know if it's going to work yet. We'll see. So anyway, that's a long answer to a short question.
I guess I'm touching upon one of the caveats. You talked about the SHSY cell line. Is that a glial cell line?
It's a neuroblastoma.
Neuroblastoma. So is it like a precursor of the PD area, the substantia nigra area cell?
No, it has nothing to do with that.
I just-
It is dopaminergic like. So I guess you would say, it's not. I mean, it's a common neuronal cell line that's used in many different studies. And it can be differentiated into a cell which will have some of the characteristics of the dopaminergic neuron. And because the dopaminergic neurons are the cell type that's most affected in Parkinson's disease, we thought that this would be a useful model. Of course, the hope and the goal going forward is to use more relevant cell models such as an iPSC development system or using, you know, neuronal cells or doing it in, steroids or organoids, which I'm sure you've heard about. There's some challenges with that, and I think I talked to you guys about that and alternative ways to assess the phenotype. But that is a significant concern is the cell type and how relevant it is. And these are all cancer cells, and all cancer cells are just mucked up in and of themselves. And they have a unique genetic background, and that unique genetic background can affect the set of genes that you can identify. So for people here who are geneticists, you'll know about things called what's called synthetic lethality, which is essentially that the importance of any particular gene depends on the genetic background in which you assess that importance. And in cancer cells, that's totally screwed up. So that's a big caveat to research. Yeah.
I'm sort of thinking about strategies for, so you came into this and mentioning that there's hundreds of thousands of chemicals that we need to be thinking about. And I've been sort of mulling over, like, is there a strategy from sort of jumping from testing chemicals kind of one at a time and in these cell populations to basically scaling this sort of like potentially thinking about all the chemicals, everything, right? As sort of like a screening chemicals. Now, I guess you could just screen to see if it kills cells,
Yeah.
but I don't know if there's how one integrates kind of like a minimalistic panel maybe across large numbers of chemicals.
Yeah.
Have you thought about how to sort of like make these up? It seems like ABCC1 probably changes
Everything.
almost all the time in almost every experiment, right?
Well, no, it's certainly in our case, and then, but I think that's also because we were choose the examples again for metals and metalloids.
Okay.
And so it's pretty particularly involved in those. Anyway, so yeah, I have been thinking about that. There's different ways to do it. It is a challenge to identify. That's kind of what I was saying with the Tox modules. And once we get to that, then maybe we could focus on those modules, and then, rather than assessing everything, we can just like pull, like, I guess I'm thinking of them as like indicator genes. Like if we see this gene, that's an indication that this pathway is probably important. And then, and hopefully if we accumulate enough information, then we can come up with more rapid ways. Because obviously, this is the opposite of high throughput, right? This is super low throughput and
Per
it's
chemical.
not going to get at that question. So, yeah. Yeah.
Have you looked into the DCAS9 technology at all? Or
Well,
maybe?
sure. So he's talking about an alternative approach, which is DCAS. So that's called dead cast. So that just means that you're inactivated the cutting, and then then you can mix. It will still target, but it won't cut. And so then you can put all kinds of different effectors attached to it, and so you can turn on genes, turn off genes, modify epigenetics. You can do all and localize. So there's all kinds of cool things to do, and some of the work we haven't talked about is one of the things we've done is activation screens, which is sort of turn on all the genes and see which things we identify. Some people argue that CRISPR-I is better because you get a better knockdown, but it's a knockdown rather than a knockout. So I think there's kind of I don't know. I mean, there's two camps. You're in this camp. It's a hell of a lot easier to make a knockout library than it is to make an inhibition library, because it's really challenging to know which of those, because you target the transcriptional start site, and it's really challenging to figure out where to put your inhibitory you're putting an inhibitory protein in there, and it's really gene to gene dependent, like where it's gonna work. Anyway, but so, yes, I think, but we arbitrarily chosen this. But I don't think I have a lot of things off. Well, thank you so much for taking the time, and please, if you ever are interested in using CRISPR in whatever your area of interest is, I'd be happy. We are very collaborative, and I've worked with people from all walks of life, and I have actually have papers published in plants. If you look me up, you will find Arabidopsis. It actually turned out to be like one of my most highly cited papers, so I actually probably just made a mistake. I have a research scientist who was from France. He worked in my lab for several years, and he was an Arabidopsis guy, and and so I said, yeah, whatever you want to do is fine. So that was back in the day.